This experiment is about NMR spectroscopy. It has two parts. One part is the dry lab. So in that you wanna learn about the theory of the NMR, how to interpret the NMR, and you will get the problem sets. So in that problem set you will figure out the structure of the unknown compound. The molecular formula will be given, IR will be given, and NMR spectra will be given. So you're going to interpret both of them to figure out the structure of the compound. Next part of the experiment is going to be the wet lab, where you are going to record the NMR of an unknown and the IR of an unknown. And then again, you're going to interpret to figure out the unknown. So what do you need in order to do this experiment? Of course, NMR. So this is the NMR machine. Here we are going to inject the sample. That's the inlet and that's the outlet. Here the droplets will come out. So you would see when I will inject the sample. You would need DI water, unknown sample, acetone, and the waste beaker. Then we also need the IR to record the IR spectra of the unknown. Here you can see I am shimming the NMR instrument. So basically shimming is a procedure to generate homogeneous magnetic field. And this is important to reduce the noise in the instrument when you will record the NMR spectra. And if the instrument is shimmed properly, then the resolution is better. So here I'm going to do that. It takes about 1.5 hours to 2 hours to shim the instrument. This is 45 megahertz instrument. So this instrument takes about, as I mentioned before, 1.5 hours to 2 hours of shimming. But when you will go to the high resolution instrument like 400 megahertz magnets or 800 megahertz magnets, they need a really long time for shimming. So by the machine you would see these filters, microfilters. They have the size of 0.22 micron. And I have filtered unknown and acetone using these filters. The reason to use this filter is if you have some impurity in the solvent. So they should basically be filtered so that you can keep the machine clean. The inside loop of the machine where the sample goes and comes out, that should be clean. I'm going to add the TMS, tetramethylsilane, to the unknown before injecting the sample. From the dry lab, you already know the purpose of adding the TMS. TMS, the chemical shift is 0 ppm. Okay, so we're going to set it at 0 ppm when we'll process our data. And all other peaks in the unknown or any other compound will shift with respect to TMS. So that's how we can have, different protons will be at different chemical shifts. Now it's time to inject the sample into the NMR machine. So I have a syringe. This syringe will be used to inject the sample. This is the inlet and that's the outlet. Make sure you have a waste beaker under the outlet so that the drops should fall in the waste beaker. Now I'm going to fill 0.2 milliliters of the unknown into the syringe. I've just filled the unknown and it should be 0.2 milliliter. And then if you have any air bubble, you need to remove it. You can just tap the syringe. If you see any air bubble, pull this down, try to remove. So basically, it should not have any air bubble in the syringe. I've already washed the syringe with 0.2 milliliters of the unknown. So this is the one I'm going to inject into the machine. Now I'm going to inject the sample. So you can count 10. The 10 drops should come out in the waste beaker. So I've injected the sample now and you have seen the drops were coming out into the waste beaker. So now I'm going to run the sample. I just injected it. So you should see that on the computer screen, you should have run one pulse. In this mode, basically you are going to collect the data. And you can name the run. This time it is unknown for me. And you can change this number, scan. So I will just keep it at 9, for now. Other than that, you are not supposed to touch any of the parameters. And I'm gonna here hit start run. You can see the scans are there coming here. So scan number 1, scan 2, and that's how it will go until all the 9 scans are completed. So the run is complete. Now I am going to download the data, as you can see here. And this is gonna open directly into the MNOVA software. This software is basically used to process the data. We can't interpret right now, but then now I'm gonna process it so that you can interpret and figure out your unknown. I'm going to zoom in. I just need this portion of the spectrum. As you can see, this one, the rightmost peak, this is the TMS. So I've added like 2-3 drops, but looks like it was more for the for the amount of unknown sample. But we know that this is the rightmost peak is basically the TMS peak. So we need to now reference it. So here you go, reference. Here I'm gonna set it 0, and you will see the shift now. So if you have noticed that all other peaks, they are shifted in reference to the TMS, which is at 0. A little bit zoom in again. Now you can see this is the clear spectrum of your unknown. Now let's learn the more about the analysis, or more about the processing of the NMR data. So I'm going to integrate now peaks. So go to analysis, click integration, pick manual, and then I'm gonna pick this singlet here because I know this is due to 3 protons. So if you know, it is very easy to pick the peak and then integrate it. You can also have the automatic integration, but then sometimes the automatic integration is not very helpful when you interpret the results. So I'm gonna interpret and show it to you here. So this one you can only do if you know the structure, right? So if you don't know the structure, you can't integrate it. So I'm gonna right click and hit edit integral, and I'm gonna do it basically 3 and enter. You would see that this basically is peak for 3, right? And this will automatically adjust. You can see this one integration right there, and similarly the integration for this one right there. I'm not going to integrate this peak because I know that this is due to TMS, so this is not the part of the structure. So you just saw that I have integrated, right? So this compound in total have, as per the integration, 8 protons. So you can see this is for 2, this is for 3, and that's for 3. Let's say if you want to increase or decrease the height of the peaks, right? So for that, you would use the wheel, which is given on the mouse, and you can move it either up or down in order to increase the height of the peak. You can see you by doing so, you are increasing some noise level as well. So I'm gonna decrease the height. Now this looks much cleaner, correct? Now the next thing comes the phase correction. That means if you want to make your peaks a little bit more symmetrical. So go to processing, hit phase correction, and pick manual correction. Then this window will pop up. And I'm gonna pick one peak at a time. So I'm gonna click it on the window, which is right there, click it there, and you can see the control comes here. I will drag it and bring it to, let's say, this peak. And then I'm gonna again go here, and when I will click the right button on the mouse, with that I will go up or down. So you can see the change in the peak. You can see the change in the peak right there. Right? It's going, a little bit symmetrical. Right? And for very minor correction in the peak, you can use the right button of the mouse. So you can see slight changes going on. Then I will bring the control here, and I'm going to do the same thing. As I've already mentioned, this spectra is pretty clean, so you do not need much phase correction here. But if the peaks are not symmetrical, then yes, you will need it, right? And then this is the TMS. So basically, this is the spectrum after processing, which you can interpret of your unknown. Now, in order to type it, just hit T for text, and the control will come here, and you can write unknown sample or whatever is your, you want to write it, and drag a little bit. And that's it. It's ready for printing, and you can have the spectra ready. So this is basically the print of the NMR spectrum. Now the waste will go to either to the halogenated waste or to the non-halogenated waste. Your instructor will let you know where to dispose it off. Once you are done with the injection of the sample, make sure you clean the syringe with the help of acetone. And then you are going to put it next to the NMR machine so that other student can come and inject the sample. Now I am going to record the IR spectrum of this unknown. I will place few drops of the unknown on this metal bump and record the IR. I will skip the details here because you already know how to record the IR from experiment number 6. And our main focus is basically to record the NMR spectrum in this experiment. IR and NMR spectra will be provided to you for interpretation.